Clean the working area with appropriate disinfectant solution. Using the match, light the alcohol lamp. Sterilize the inoculating loop in the alcohol lamp until red hot. Allow the loop to cool. Always remember to apply aseptic technique before and after opening the plates. Pick an isolated colony from the agar plate culture and spread it over the first quadrant, approximately 1/4 of the plate, using close parallel streaks. Sterilize the loop by heating it until red hot and allow it to cool. Turn the plate 90 degrees and lightly sweep the loop 1 to 2 times through the inoculated area, then streak into the next quadrant without overlapping the previous streaks. Again, sterilize the loop by heating it until red hot and allow it to cool. Turn the plate 90 degrees and overlap the previous area once and streak into the next quadrant. Flame the loop for the last time until red hot to sterilize it. Label the clean glass slides before starting the procedure. Using a match, light the alcohol lamp. Allow the slide to pass through the flame 2 to 3 times. Repeat this procedure on the other side of the slide. Heat the wire loop until red hot before using. Always allow it to cool to avoid melting the agar. Always remember to apply aseptic technique before and after opening the culture plate. Pick an isolated colony from the culture and transfer it to the glass slide, creating a smear. Heat the wire loop until red hot to sterilize it, and set the glass slide aside. Repeat the procedure on the second glass slide and pick another isolated colony. When both the smears are set, let the back portion of the glass slide pass through the heat for heat fixation. Once done, turn the flame off. Place the slides on the staining tray and pour crystal violet as their primary stain. Rinse the slides with distilled water. Remember not to touch the very smear because it might fall off. Apply mordant, which is the grams iodine, and wait for 1 minute before rinsing with distilled water. Do Do a rapid decolorization using alcohol until excess stains are removed. Immediately rinse with distilled water after decolorization. Pour the counter stain, which is the saffronin red, and wait for 10 seconds before rinsing with distilled water. Wait for the slides to dry. Examine the stained smears under OIO. Start off by labeling a clean glass slide. Heat the inoculating loop until red hot and let it cool. Place 2 drops of hydrogen peroxide and observe for effervescence. Start off by labeling a clean test tube. Light the alcohol lamp with a match. Always practice aseptic technique before and after opening the tubes. After applying aseptic technique on the EDTA tube, pipette 5 drops of plasma. Close the EDTA tube and apply aseptic technique on the test tube before transferring the plasma. Heat the inoculating loop until red hot and let it cool. Perform aseptic technique on the culture plate. Scoop out a generous amount of an isolated colony. Perform aseptic technique on the test tube before transferring the colony. Heat the inoculating loop until red hot to kill all the bacteria. Heat your inoculating wire until red hot and let it cool. Perform aseptic technique before getting an isolated colony. Start inoculating on each of the test tubes. Remember to perform aseptic technique before and after opening the test tubes. Do not reheat the wire loop in between inoculation. Reheat the wire loop until red hot to kill all bacteria. Lastly, label the test tubes according to their corresponding tests.